Okay, next speaker is Sergio Rotstein, who will talk a bit about the HELM project with Pistoia and also Chemaxon's involvement.
So, all right, thank you for inviting me to speak. It's good to see so many old friends and make lots of new ones too. So thank you. As you see the title of my talk, What About the Big Guys? This is not a talk about Doug Drake. It's actually about the HELM technology. The talk's going to be divided into two parts. The first one I'm going to talk about, it's more Pfizer-centric. It's why we needed this, how we created it, what we do with it. Then the second part, I'll switch hats and talk about Pistoia Alliance and what we're doing to externalize the technology, make it available to everybody else. So first, the Pfizer part. This was born out of a strategy decision at Pfizer. Pfizer had traditionally been a small molecule target-based drug discovery company like many. I'm good, thank you. Thank you, Papa. And at one point, the company decided that it wanted to diversify into the biotherapeutics realm, and Pfizer likes to go big, so it's like we're going to become a top-tier biotherapeutics company, and we'll do it in 3 years. Why not? And so the informatics infrastructure, of course, was going to take quite a bit of effort to catch up because it's quite a different type of research from what we did traditionally. So the Biomolecule team, which is my team, had the goal to remediate the infrastructure, basically to enable this kind of work. And these are the primary areas we needed to work on. First of all, registration, as all of you here know, it's really the enabler of everything else. You need to register your molecules so you can have them in the database and associate all the data with them. And then everything downstream, visualization, analysis and design, and workflows. And as we got to work more and more on this, and we got to talk with our colleagues and some of you even, it quickly became clear that Pfizer was by no means the only company doing this. And as it turns out, really all of the companies, big and small, do tend to work in all sizes of molecules and all that kind of space. So everybody was kind of on the same boat, we found. And so that's when we got involved with the Pistoia Alliance, having the goal to facilitate the release of the whole technology, the goal being that we can have a standard by which all of us can exchange data, because as you know, it's a very highly collaborative industry now. And then also for the companies that perhaps were a little behind from where we are at Pfizer, why reinvent the wheel? If you can use it, if you can benefit from it, that's great. So first, a little definition, because I think we probably made up the word biomolecule. I don't think it exists. So for our purposes, we call anything that's not a small molecule a biomolecule. That's totally wrong, I know, but you got to call it something, right? So I'm just showing you a few examples here: peptides, proteins, oligos, antibodies, bioconjugates, all these crazy things, and then vaccines, which is basically a soup of all of those. And we wanted to eliminate what we've been calling the biomolecule penalty. And what that means basically is that if you were a biotherapeutics researcher at Pfizer, it was simply a lot harder for you to get your work done than, from an informatics standpoint, than if you were a small molecule researcher. That's not fair, so we needed to remediate that and make these entities what we call first-class citizens of our tool portfolio. And so let's focus on the actual problem at hand, and this was covered a little bit earlier today. But what we were facing is the following. From a small molecule perspective, if you are the old Pfizer or any small molecule company, you have so many tools, many of them from Chemaxon and many from others, to work with these. It's easy. You can draw them, you can search them, you can do all these things. It's kind of a done deal. Never done, but pretty much a solved problem in some respects. On the other end of the spectrum, if you are working in target space, genes, proteins, etc., you also have a nice rich set of tools to work with that. Your blasts and your FASTAs and whatnot. When it comes to sequence, storage, searching, finding, clustering, all that, there's a lot of solutions in that space. But we ended up stuck right in the middle here. And so the kinds of molecules that we work in, they are too gigantic to be small. So you don't want to use Marvin's sketch, to draw a protein, I don't think. But at the same time, they're just too weird to be sequences because they have all these attachments and weird hybridizations and all these things. So they end up breaking both kinds of tools. And that's really the root of the problem, why we got to work on this stuff. So what we really wanted to come up with was a fit-for-purpose structure representation, something that could scale well with the complexity of the molecules at hand. And so just to exemplify it a little bit, and this is where we got our start, which was in the oligonucleotide space. If you're dealing with these short oligonucleotides, there is no doubt that you can draw this if you have time. You could do this, and in some cases you might even want to. But by and large, people doing oligo research, they don't think in these terms. They think more in sequence space, more in these cartoon little depictions that I'm showing below. So this is an example of how we would represent an oligo or a peptide, something kind of short. But at the same time, even if you solve this problem, then you try to scale up. Even the cartoon I showed before, you don't want to use this for a whole protein. That doesn't make sense either. So you want to be able to now go and represent things in your more normal sequence space, like an example, heavy and light chain for an antibody. But then that's not all either, because now you get into the realm of bioconjugates or multi-component structures, and people like to think of them in their individual components. So a typical example, is your antibody-drug conjugates. And so depending on what you're doing, you could imagine and put it in the context of doing SAR for this kind of work. A lot of times what you're doing is you have a fixed antibody, and what you're varying is sort of the conjugate, the payload, if you will. So when you're doing SAR, at that point, you really don't care about the antibody so much. You just want to look at these guys and do normal SAR around those. On the other hand, we also had situations in which the linker payload remained fixed. It was the antibody scaffold that varied for metabolic reasons and other things. And so maybe now you don't care about this, but you want to look at that. And so there's this notion of components, and you want to be able to focus on the one that you're interested in, and you want the system to understand that these are individual components. So that brings us to HELM, the Hierarchical Editing Language for Macromolecules. I know, I don't like the name either. So we had a few goals in mind here, and I'll just walk through them a little bit. The first one is hierarchical. This speaks to what I was just saying about multi-components, and the idea being if you have a multi-component or complex polymer type of molecule, it is made of simple polymers, and simple polymers are made of monomers, and monomers are made of atoms. And you want to be able to kind of zoom in or out depending on what you're trying to do. We wanted it to be extensible. We started this work in oligonucleotide space, but we quickly expanded into peptide space and then protein antibody and such. But it's not done. I forget the gentleman I spoke with was interested in doing, carbohydrates and that kind of thing. There's plenty of other types of polymers, and so you want it to be extensible and be able to add whatever crazy polymers your scientists want to throw at stuff. We wanted it to be reasonably comprehensive, and what I mean by this is that a lot of structures can be, even though you can represent them structurally, that doesn't always paint the whole picture. So an example is, as I'm saying there, it's oligonucleotide hybridization. You know, and we were having at some point triple-stranded oligonucleotides with some chemical attachment in between them. It's very weird stuff, and you want it to be representable. That's the whole point. The desirable was for it to be canonicalizable, meaning just to make it easier for quick checking for uniqueness. And then, if at all possible, we wanted it to be kind of human readable to make it more portable, and the kind of thing where the computational folks would really kind of want to look at this and know what it is. And just think of smiles. Smiles, you could say, are not really that legible, but anybody who's worked with them for any time, you can read those structures like that and really interpret them pretty well. So just to do an example, and this is not going to be a HELM tutorial or anything, so I think you all look sleepy enough as it is. But just to give you an example, this is what a peptide looks like, and I'm showing the HELM notation, and just a couple of points of note. When you have an unnatural type of monomer it's represented with the brackets here. And usually when you have unnatural monomers, you can quickly convert this into what would be the natural equivalent. I cheated a little bit. I threw the X there because sometimes you do have monomers that are just not, they don't have a natural analog, so you have to represent those somehow. So that's HELM kindergarten, and then this is HELM graduate school. In here you have basically an oligonucleotide with sort of an unnatural base that's connected through a chemical linker to the second amino acid of a peptide. Good? Got it? So that's what the HELM notation looks like. And believe it or not, I think in a couple of weeks, you could read this and know exactly what that is. It's kind of crazy. Actually, I should ask Akos, or some of the people who've been working on it are probably pretty good at it in ChemAxon. And this is just like a little cheat sheet to tell you some of the basics of the representation and how it works. Now, since you saw how these polymers are represented through monomers, for each of the monomers that you are representing here, you obviously need a way to define what they are, and you need to have some flexibility, particularly for unnatural monomers, for people to define each one as they wish, give them the names they want, et cetera. So I'm just showing what alanine looks like in our monomer database, which defines everything. And won't dwell too much on it, just to say that you have your backbone, and essentially has the attachment points where you can either grow the backbone or attach whatever spinach you may want to attach to it. And if it's unnatural, it would have a natural analog, et cetera. So that's more or less how behind the scenes the system knows what the chemistry is behind each monomer. So if you can't get enough of it, you can go and read the paper. And actually, in the Pistoia Alliance website, we're now publishing the paper free for download. So it's been arranged with ACS so that anybody can download it. So feel free to do that. And just to take a minute to talk about, now we know what HELM is, how are we actually using it within the company? And so at the core of everything is the drawing tool where you can actually create these molecules. And so it's you can think of it as Marvin or ChemDraw or whatever, but just kind of like a Lego block equivalent. And this is how you build your molecules. You can drag and drop and build a chain that way. You can enter a sequence there, and it'll create it for you. So that there's various ways to input the information. And just a little peek at our monomer database here. And it's just to make the point that at Pfizer, the Monomer Database is a company central monomer database. And so we have the concept of curation. And if you're a super user and your monomer, you want to create a new monomer, it's not there, you can go in and create it yourself. If you're not a super user, this little register button is not there, and you can go and say, please add this monomer, and curator gets an email, and they can add it, and then it shows up in everybody's canvas, so to speak. So that's the drawing tool at the center of pretty much everything. Then here's a look at our registration tool that leverages it. Two examples here that depict kind of how it scales with the complexity of the molecule. So the simple example here is just like a cyclic peptide. So here it's running in what we call peptide mode. But then if you switch it to protein mode, then you can work in a more sequence-oriented fashion like that. So it's just two examples of two different biomolecules that you can register. And then on the other end of the spectrum, again, it's the data's in there. What do you do with it? What I'm showing here is a tool called pFred. And pFred is a tool for oligonucleotide library analysis and design. So what you're looking at here is in the analysis mode, where basically you have all your oligos in the context of their data and your IC50 curve fits, and you can do various activities. And you can do a lot of standard stuff. You can call Spotfire, CCT services, which 2 people in this room know what those are. But it's a computational service that you can calculate properties, et cetera. You can do a lot of the standard things that you can do with an analysis tool. But then it has a design tool, and it's a little hard to see, but you can basically enter your gene of interest, and it'll burn through a library of oligos. It'll create them all, and then I'll try see if that works. Aha, that worked. You can now decide all of the different features that are good or bad for you, and it'll apply those features to the calculations, and then when you're done, you get your library of oligos. I talked about some workflow tools that I won't mention too much, but just to say that when you're done with creating your library of oligos, you could actually click a magical submit button, and then the groups that the chemistry groups will actually synthesize your library and register it and do all of that behind the scenes. So there's some integration with workflows there. Okay, so I'm now going to change hats. I'm Pistoia Sergio. See, even my email is different for the second half of the talk. I feel like I should change places. I'm a different guy now. What's that? Deeper? I can't do that. It's hard enough. So I mentioned this at the introduction, but the project aims were to establish HELM as a representation standard with two main goals. One of them, use it as an exchange mechanism for data, and the second one, hopefully reduce software development costs so that people don't have to sort of reinvent the wheel. And so in terms of participation, it's actually been kind of a fun project in that there's so many companies that got involved, and it's been great from that perspective. And just about everybody you see there has had some level of hands-on participation, which has been very, very cool indeed. And we've now wrapped up what we call the first phase of HELM. And the first phase had to do mostly with taking HELM out of Pfizer, where it was as a notation language and set of tools, and into the public domain. And that involved a certain amount of work. Ultimately, what we need to get to is a situation where the software attached to the HELM notation standard is out in the open source, has no links to Pfizer whatsoever, and is basically progressing as a software package, just like any other open source project would, hopefully with people contributing to it. And at the same time, the definition, the language itself, can continue to evolve in some managed fashion because it can do a lot, but it can't do enough yet. There's plenty of problems that are not solved, so the language needs to continue to evolve, and hopefully as an established standard system that maybe people can use in their systems, in their companies, and then vendors could hopefully either use the technology, adopt it, and improve it, or at least support it for input-output type. So this is a super busy slide, but it's been a busy project. So just to tell you where we are with it, the source code is out. And basically, we have an open source repository for it. We've selected GitHub for that. We spent a lot of quality time talking about open source license decisions, and we ended up choosing the MIT one, which, if anybody's familiar with open source, MIT one says, I'll summarize it. It says, we're not responsible for anything, and you can do whatever you want. That's about what it says, and very, very low on the legal language. So the point of that choice is to maximize adoption. We don't care if vendors, if Chemaxon takes this technology, adopts it, and turns it into something profitable, that's great. But our goal is satisfied. We maximize adoption across the industry. And then, of course, people making contributions to the project do need to make sure that the project owns their contributions. So that's what this contributor license is about, which we're using Apache. And we've said some of the basics, a governance structure and whatnot. The next one here is one of my least favorite words, but the source code has been de-Pfizerized. It feels like something is taking it from my chest or something, but it's now deposited into GitHub, into the repository. Chemaxon, who's been the hardest contributor to this whole project, created a version of PME, the molecular editor, in applet form, which enables now everybody to try it without having to install software. So it's been very, very cool. And we're doing some of the associated things like a user guide, a video tutorial, this kind of thing. Very recently, I was very happy to see a prototype from Accelris that can now read and write in the HELM standard on Pipeline Pilot, which I thought was very cool. And then we have just deployed openhelm.org. It's a website. You're welcome to go to it, and that's where you can find out more about the project, download the paper, check out the applet, get to the source code, et cetera, et cetera. And as of all of maybe 48 hours, that the website looks to be live and up and running, so it's exciting. And then finally, we've just kind of tried to do, in a scrappy fashion, get the word out as much as we can, participating in forums such as this one. So what's next? We just finished phase 1, and phase 1 was a lot of work for a lot of people. And now really is when we should get it to a point where people can benefit from it and hopefully continue to evolve the standard. So this is where we want to get to. So now we're in the process of starting to meet about, okay, what's next? How do we now go from, okay, it's out, now how do we improve it? How do we evolve it? And so that's what phase 2 will be about, really, the next stage of the project. And ideally, you know, we really mean we want to maximize adoption. So if you're in a company that is struggling with this notion of biomolecule representation, registration, et cetera, and you have your own software development resources, et cetera, they should go and look at this and try to use it. Why not? If you don't have your own resources, you can hire Mike over there. There's plenty of people who'd be happy to work with you on it, and I definitely encourage that. And again, with the vendors, all vendors, it'd be great if can at least adopt the standard, if not the technology itself. So going down, this is actually, it's an acknowledgement slide, but it's actually my favorite slide of the presentation. And the reason is that I have never been in a project where like every company is there. There's like companies and vendors and resellers and all these people involved. Every single person working on a volunteer basis, meeting a ton, working their tails off in an incredibly collaborative fashion. So it's actually been a lot of fun, and I would encourage anybody to get involved just because it's really interesting, right? We're all competitors and all that, but in this project, we really worked very well together, and a special thanks to our excellent project manager, Claire Bellamy, because despite what I just said about how hard everybody was working, if it wasn't for Claire cracking the whip every now and then, we'd all go back to our day jobs and kind of disappear, right? And then a very, very special thanks to Chemaxon. Basically, this de-Pfizerization that I mentioned before is a lot of work. You know, it's a system deeply integrated with our infrastructure. And let's face it, we and Pfizer have plenty to do. We can't do the de-Pfizerization. It's just something we don't have the resources to do. But Chemaxon really stepped in and did all this work and did an excellent, excellent job. And actually, on that note, I'm going to go off-road a little bit and ask Alex if he could come join me here for 2 seconds, because we have a... See, this is Pistoia Sergio now, so I actually have a present for Chemaxon on behalf of the Pistoia Alliance. Yes, I just get the big cheese, you know, you can do whatever you want.
That's very nice of you.
So yes, so it's hard to read from there, but the Pistoia Alliance hereby recognizes Chemaxon Limited for outstanding contributions to the Pistoia HELM project, May 2013. So our special thanks
I'm not going
to
to
you
accept
guys.
it. I'm going to ask Agros to accept it. Yeah.
There you go.
See,
Thank you. That's
I can't
fantastic
personally
job.
accept that because I'm a member of the steering committee,
Thank
which
you.
means I'd be giving it to myself,
You know,
even though I didn't know about it.
that's the insider. So that's it, and thank you
very
so
much,
much for your attention. Thank you.
Guys.
You get a fabulous box. I just don't want Brock to ask me anything. That is a horrible way of doing it.
Hi, that's very, very nice. I mentioned this at this point at the Pistoia meeting before, but my guess is I would speculate that in the area of complex biomolecules, the patent offices can no longer verify prior art. So unless they get HELM, that's going to get worse and worse and worse.
Yeah, I mean it's a huge widespread problem that everybody has. So
I mean, have you talked to the patent offices?
we've talked about it, and I guess you know, the drug companies usually from a technology standpoint, tend to be a step or two ahead of the curve with respect to the patent offices and such. And so this is just beginning to percolate into sort of the space of the biopharmaceutical companies. It would be natural for those who sort of end up dealing with the end products to follow suit, but I would say it's still pretty early. So no, we haven't had any specific conversations with like the U.S. Patent Office or anything, but you saw the INCHI Trust, we talked with NIH, a lot of the people who get involved in the setting of standards and whatnot. So I think they're the kind of the right people that are having those conversations, but nothing concrete yet.
Have you looked at glycoproteins during your discussions?
Sorry?
Glycoproteins.
Glycoproteins,
Because there are two issues here. One is oligosaccharides are typically tree-like structures, not so much polymer structures. And the second is glycoproteins very often exist in glycoforms. That means the same glycosylation site carries in natural abundance a mixture of carbohydrates.
So the stuff that we've been working on is sort of our state-of-the-art as of the paper publication and writing. So it's a bit 18 months or so behind. But we do indeed work with that. And just to generalize the problem, there are representations that are simply unknown or ambiguous. And glycosylation is a great example of something. But it happens also in the antibody-drug conjugates, where you know what your structure is or your sequence is, you know what your linker payload is, but you're not sure where it's going, or you have maybe a stoichiometry idea, but that's it. So the next generation of the representation starts to drill down into ambiguity, or how you deal with stuff where you can't go down deeper. You know, the hierarchical representation where you can't go deeper because you just don't know. So it's a good question, and it's a problem that we've been looking at.
In that same vein, glycosylations are also used for protein stabilization. Do you represent that in the registration, or that's arbitrary as well?
If it's a concrete, it's just chemistry. So if it's a concrete attachment and you know where it is, you can represent it. It's the ambiguity that gets weird because you don't always know.
I apologize if you mentioned this, but just for clarification, you've shown how you enter these macromolecules. You've got different options.
Yeah.
So how are these stored? Do you store that string, or do you store the chemistry behind that string, and can you search it?
Yeah, you store that string, and because the combination of that string and the monomer database that contains the chemical representation, between those two, you can either export things as plain sequence, you can export them as smile strings, or anything in between. Searching, we can do searching, sequence-based searching and chemistry-based searching. But a lot of the real questions that people want to ask, give me every bioconjugate that has this particular sequence moiety and this substructure, that kind of complex thing, we're definitely not there yet. And that's actually one of the things we're hoping, putting it out in the open source, that other people will be able to answer those, sort of to tackle those difficult problems. Okay. So, thank you.